In this video, we are going to see what is monoclonal antibodies. What are monoclonal antibodies? The antibodies of uniform specificity produced by a clone of a B cell against a single antigen determinant or a hapten are called monoclonal antibodies. Monoclonal antibodies are produced by hybridoma technology developed by Colher and Milston in 1975 at Oxford University. On contrary, some of total antibodies produced by clones of different B cells against all antigen determinants of a multivalent antigens are called polyclonal antibodies. Following natural infections or vaccination against a pathogen, polyclonal antibodies are produced in vivo, which are measured quantitatively by various serological tests. How to produce monoclonal antibodies? Hyperimmunize a mice against an antigen. Sacrifice mice ethically and collect spleen. Seek the spleen followed by trypsinization to obtain single B cell. Seed cells in vitro in a suitable culture medium 96 well plate by limiting dilution. Examine well showing a single cell. Screen supernatant from such wells for production of antibodies of desired specificity. Expand positive well. The problem is the splenic cells are mortal and will die soon due to apoptosis. This is the way to produce monoclonal antibodies. What is hybridoma technology? A technique to generate continuous supply of monoclonal antibodies. Take advantages of unlimited growth capacity of myeloma cells and cellular machinery to produce antibodies. This is the strategy to generate monoclonal antibodies. The antigens are injected, the spleen cells are isolated, and it is their plasma cells and myeloma cells are hybridized, and they are selected to hybridomas and they are cloned, and the monoclonal antibodies are selected. This is the strategy to generate monoclonal antibodies. Hybrid cells. Normal splenic cells are fused with cancerous cell line. Fusion is accomplished with PEG, polyethylene glycol. The new hybrid cell exhibit properties of both cell types. Unlimited growth from myeloma cells. Secrete monoclonal antibodies from splenic cells or secrete cytokines. Hybridomos are hybrids between a non-transformed antibody producing B cells and a transformed cell myeloma that can grow continuously in culture. The normal TR-B cells, cancerous TR-B cells, nucleus of normal lymphocytes, nucleus of cancerous cells, random chromosomal loss, possible fusion products. Myeloma cells from immunized animal plus myeloma cells and fused plasma cells and hybrid cells, fused myeloma cells and unfused myeloma. These are the possible fusion products. Pathways of synthesis of purine nucleotides. There are two types of pathways, the de novo pathway and the salvage pathways. Myeloma cells. This cell line cannot secrete any immunoglobulins. This cell line is deficient either HGPRT or TK, which are enzymes of salvage pathway. Hence, salvage pathway is not possible. Cell line cannot survive in selection medium which contain aminopterin. Aminopterin inhibits de novo pathway. Salvage pathway is not possible due to HGPRT or TK deficiency. The TK is a thymidine kinase deficient. So the cells passes through the salvage pathway. Possible fusion products. Plasma cells from immunized animal, myeloma HGPRT deficient and Ig deficient. Senescence can use salvage pathway, no senescence, hat medium not possible. Hat medium is used to select for growth of hybrids and against the growth of parental myeloma. Spleen cells producing antibodies from mouse immunized with the antigen A. Myeloma cells lacking antibody secreting the antigen enzyme HGPRT. This is the process of the monoclonal antibodies which are taken. Immunization of mice for monoclonal antibody production. Intraperitoneal immunization aesthetically isolates spleen cells. Animal is immunized with antigen, spleen cells are isolated. Day 0.50 UM of AG is complete funeral ascendant. Bleed animal and test reactivity to antigen. Final boost of 10 antigen. Fusing spleen cells with myeloma purifies spleen cells and fusion takes place. Selection of hybridomas. Selecting clones. Feed wells with new hat medium. Feed cells twice a week. After about 2 weeks, test supernatant of potential clones using ELISA. Retest positive clones in 48 hours. Expand clones in new 96 well plate with HT, not hat. Retest suspensions. Expand in 24 well plate. Switch to normal medium. Retest. Expand in 4 well plate. Cryopreserve. Enduring monoclonality. To ensure monoclonality, subclone hybridoma serial dilute at 1 cell per 3 wells. Uses spleen cell as feeder cell. Feeder cell concentration 1 into 10 power 6 of cells milliliter. 96 antibody production. Large amount of antibodies can be produced using animals. Alternatively, bioreactors are used. Cells are cultured in hollow fibers. Fresh media and waste are recirculated. High concentration of AV production produced in cell compartment. Antibodies collected at different time points. Others roller bottle, microcarrier, membrane bound cell culture process. Large amount of antibodies in large amounts. Inject clone, collect peritoneal assets. Advantages of monoclonal antibodies. Homogeneity binds to same epitope with uniform affinity, high specificity, target oriented, small quantity of antigen is required, consistent, limitless supply of specific reagent, more easily tested for cross reactivity. Disadvantages of monoclonal antibodies. Highly specificity affects use of diagnostic when there is minor differences between strains. Limited sensitivity. Average affinity is lower than monoclonal antibody. Immune rejection. Expensive, time consuming, and the scales. Uses of molecular antibodies, autoimmune diseases, cancer therapy, and also in metabolic disorders and infectious diseases. Metabolic disorders, targeting secreted fatty acid binding proteins, new treatment for type 2 antibodies. Infectious diseases, racibozumab. Thank you.